My name is Andrew Bowe, and I am on the Hutch Connect and Science for Staff committees that bring you this series. Please visit the Science for Staff page on Cinenet, where previous lectures can be viewed and our upcoming presentations are listed. Our featured speaker today is Dr. Sam Minot. Sam is a staff scientist in the Vaccine and Infectious Disease Division. He is currently the Bioinformatics Core for the Microbiome Research Initiative. Sam received his bachelor's degree from Bowdoin College in Maine and his PhD from the University of Pennsylvania. He joined Fred Hutch in 2017 after working in both Austin, Texas and San Francisco, California. Sam has been studying the interaction between microorganisms and human health, which he will be elaborating on this morning. Feel free to submit any questions that you have during the lecture. You can submit your questions via the chat function on Zoom. At the end of the seminar, I will read the questions to Sam. Welcome and thank you for joining us this morning, Sam.
Thank you very much. I appreciate having the opportunity to speak to you all today, and I appreciate everyone joining us to talk about the microbiome and human health and disease. So I want to share with you today some of the really exciting parts of the science that I get to do. And this is really around the discoveries that have been made in the field of the microbiome, uncovering this immense, powerful, and exciting world of microbes that live on our bodies. At the same time, I also want to tell a bit of a story of technology, ways in which technology has transformed and really enabled this entire field of science. And it's changed not only the way that we conduct experiments, but it's also the way that we conduct our research, the way that we organize ourselves as scientific staff. And so I want to try to convey a little bit of both of those aspects of this field and my work in it. So a little bit about me and my perspective coming into this work. So I'm trained as a microbiologist. I am a computational biologist, and I work in collaboration with other scientists. So here, as Andrew mentioned, I'm part of the Microbiome Research Initiative, which is an initiative led by Dr. David Fredericks to enhance and support all microbiome researchers at Fred Hutch. And I do this in a collaborative way, taking on the data challenges and trying to provide solutions to a wide range of researchers in a way that can support really the complexities of analyzing data in terms that are encountered in the microbiome. So let's start here by talking about what the microbiome is. So this is a phrase that certainly has come up in the news, in the popular press, and the microbiome really refers to the collection of organisms which live on and inside the human body. Now, that's a phrase that people that you've probably heard before, and I want to unpack this a little bit more. So we're talking about microbes that are on the surface of our skin, in our mouth, in our saliva, in our gastrointestinal tract, and they belong to a really incredibly diverse collection of different organisms. So we're talking about bacteria, viruses, fungi, microeukaryotes, as well as archaea. And there's just some of the examples of these organisms are shown here. So here we can see the different types of names that are given to individual microbes. So for example, Helicobacter pylori or Escherichia coli, and the different body sites that you might find. So really different types of organisms are found in all these different locations throughout the body. To unpack this even more, sometimes it's helpful to get an idea of even just the scale. People say that there are microbes living on the human body. Well, how many are there really? Well, if you look on the surface of the skin, you may observe up to millions of individual microbial cells per square centimeter. And this may seem like a lot. I don't want anyone to, to freak out or sort of get the heebie-jeebies that there are these bacteria that are on your skin because they're incredibly small. Obviously they're microbial. You can't see them with the naked eye. You have to look under a high-powered microscope. But even just on a square centimeter of skin, if you were to lay out the bacteria end to end, a single even layer, there would be over 100-fold more than we actually observe. So even though they are on the skin, it's not like they're covering it. They're sort of scattered throughout. Other sort of body sites, when you look at something like saliva, you might find as much as 1 billion microbial cells per milliliter. But then when you get down into the stomach, it's got this high acid environment, and there's really a lot less. So down to something like thousands of bacteria per milliliter. So, 6 orders of magnitude difference. And then when you go from there all the way down to the large intestine, this is a site where there really is an extremely high concentration of bacteria, because the bacteria that are in the gut, in the large intestine, perform a number of different functions, like helping to digest food, process nutrients. They also interact with our immune system, metabolize different bile acids and detergents that we make. They really perform a large number of functions, and they're present in really high numbers in the large intestine. And this is all good. So all the numbers that I'm telling you now, these are all good parts of the microbiome. So I've used the phrase microbes, and I've thrown around some names, but I know that the taxonomy, the different types of microbes that are present is not something that's immediately obvious to sort of all people in the world. So let's talk about what microbes are. So people talk about bacteria. This is one group of microbes. So bacteria are a form of life that are, generally single-celled, but they're incredibly diverse. There's lots of different things that bacteria do. They come in lots of different shapes and sizes. Some of them, like on the left, might form these like grape-like clusters. Some of them, maybe on the right, have these tails, these pili, these sort of filamentous projections coming off of them. Really, the physiology of bacteria is incredibly diverse. And what I'm showing you up here are just a few representatives of organisms that have been really intensely studied, that are just a small selection of the broad range of organisms present in the microbiome. Distinct from that are these yeasts or fungi or microeukaryotes. And I want to draw this out as a distinction because when it comes to single-celled life, there's an incredible amount of diversity that we don't always think about because it's not visible to the naked eye. And so when you think about something like Saccharomyces cerevisiae, maybe a baker's yeast or brewer's yeast, these are single-celled microbial life forms, but they are eukaryotes like us. And so the green cells on the right are actually more closely related to us than they are to the bacteria you see on the left. And I'm going to talk a little bit more about how we fit into this tree of life in a minute. Now, this is obviously not the end of the story. So there's quite a number of other different types of microbes that are totally different. For example, viruses in the gut microbiome. Now there's obviously a lot of talk about viruses today. And I want to say that even though there are some viruses that are just present naturally in the gut, which infect human cells, there's actually many, many, many more viruses which infect bacteria. And so when we think about viruses in the gut, and this was actually the topic of my graduate work. So I spent my PhD studying the viruses in the human gut. And the first question we had is, what are they? What are they infecting? And the answer is they are almost entirely infecting bacteria. And so these are viruses that go through their entire life cycle, never even touching a human cell, just going into one bacteria, infecting it bursting, and then going off and infecting another bacteria. And this is actually a very important part of the biology and about the interconnectedness of microbes, which I think is something that is maybe a little bit underappreciated sometimes. In addition, there's actually another whole class of single-celled life called archaea. When people talk about archaea, oftentimes they talk about these extremophiles. So the microbes that are able to live at the bottom of a hydrothermal vent or in an extremely salty, so a hypersaline lake. These are the sort of extremophiles that people talk about. In addition to those environments, there are some archaea that live in the environment of the human microbiome. They're not particularly numerous or concentrated. It's not like a majority of your microbiome will ever be archaea, but they are present and they do things. They're able to perform functions that aren't performed by other types of life forms. And so what I wanted to emphasize here is that there's really a wide diversity of microbial life that exists within the microbiome. And a lot of this diversity has only expanded in our understanding as this field has developed. So I want to take all of these organisms and show you how they relate to each other. So this is a tree of life. Each of the tips of the tree, so this tree is radiating outwards from the center. At each of the tips of the tree, we have names for the different organisms that we've observed. And then you can tell how closely related different organisms are to each other based on how far apart the different tips of the trees are. I need to try to get my laser pointer going here. There we go. So when we, for example, we can look at this entire group here is all bacteria. Here we have eukaryotes, here we have archaea. And so if you want to orient ourselves to where we are one of the tips of this little tree down here. And all multicellular life, all plants and animals are really just a small part of this eukaryotic tree. And so when we ask the question, well, what is microbial life? Well, in the context of all life forms, most life is microbial. With the exception of a few tips of the tree down here, everything else that you can see here are forms of single-celled microscopic life forms that are really incredibly diverse, different from each other, and are capable of performing a wide range of different biological functions that are really distinctive between these. So these are incredibly different forms of life, and a large number of them are found within the human microbiome. So that's a little bit about, generally speaking, what is the microbiome? What is in it? I want to describe a little bit about how we were even able, as a scientific community, to observe that these microbes were present. So I want to talk a little bit about how we actually measure the microbiome. And this is going to be part of the story of how changing technology has driven changes in science, which have really driven new discoveries. So I'm going to talk here about genome sequencing. So as we think about a human, and we want to study what's in the genome of a human, we do this by obtaining a DNA sample. So, from saliva or from blood, perhaps, whatever it is, we get some purified DNA. This DNA can then be put onto a genome sequencer. So here's a collection of different machines. These are the actual instruments used for sequencing genomes. These are ones produced by Illumina, which is sort of the dominant genome sequencing company right now. And after you put this purified DNA into a genome sequencer, you then get out the sequence of the genome. And in the context of humans, this is, in a perfect world, this is a set of, a single chromosome is a word. So it's a sequence of nucleotides. So A's, T's, C's, and G's laid out in order. And for all the 23 or 24 chromosomes that you have, the genome, your genome, is really just this sequence of letters across each of these chromosomes. So this is the process that has gone about for generating genomic sequence data. And the change that's happened in this area has really been a story of rapidly accelerating technology. So when we look at the performance of these machines and how efficiently they are able to run, back at the beginning of the Human Genome Project, around the turn of the millennium, the estimate was something like it cost $100 million to sequence a single human genome. And over the course of the early and mid 2000s, we saw that this declined relatively rapidly. So the blue line is Moore's law, and that's the rate at which the cost of making semiconductors has declined. And we saw that it was related to this. So going from 100 million down to 10 million, note the log scale here on the left here. So we noticed that it went from 100 million to 10 million, over the course of less than 10 years. Now, right around the time that I started graduate school, we saw that this technology increased in its productivity quite a bit. And there was this dramatic improvement where over the course of just a few years, all of a sudden, the cost of sequencing genomes had declined by orders of magnitude. So we're getting into thousands of dollars in order to sequence a single human genome. And what this really meant was that now we had this set of instrumentation, we had this technology for sequencing genomes, which had all of a sudden got a lot more affordable. And it was a lot more practical to apply it to other purposes besides sequencing the human genome. And this is a point that I sort of like to make when people think about, well, I'm hearing a lot about the microbiome. Why am I hearing about the microbiome now and not, back in the 90s? And the answer is really because technology has advanced considerably in terms of being able to sequence genomes. That's been a huge driver of this field. And so when we look at how this applies to the microbiome, I want to really just emphasize that we can do the exact same thing for a bacterium that we can do for a human. We can take a bacterium of interest, we can isolate DNA, we can put it onto a genome sequencer, and we can generate the genome sequence for that bacterium. So when we look at what this genome is, we find that they're generally much, much smaller than the human genome. So something like 1,000 times smaller, which means that we don't actually have to sequence as much in order to generate it. It's cheaper to sequence bacterial genomes. And that the genome sequence contains all the information necessary to create this organism, just as our genome contains almost all of the information needed to generate us as an organism. And this is a point that I think is kind of, it can be a little bit surprising and fun to think about. You know, humans have a genome, microbes also have their genomes. Now, we just have the one, generally speaking. You know, every cell in our bodies, with only a few exceptions, contain a copy of the information needed to regenerate, you know, ourselves, the complete information about us as organisms. The same is true for each of the different microbes that I told you about before. So because it's single-celled life, each one of these cells that you can see here also contain this complete set of instructions. And so when you think about the diversity of all the different types of life I told you were present in the microbiome, it's also, that is reflected in the differences in those genome sequences. Each of those different forms of life have their own distinct genome, and that encodes the information needed to perform all the different functions that this particular Staph aureus might have, like pathogenicity islands or bacteriophage resistance, et cetera, et cetera. And so I really want to emphasize that our knowledge of the microbiome and our knowledge of microbes has expanded rapidly in recent years, largely as a result of the advances in genome sequencing technology, as well as a number of other things, but I think this role is hard to understate. So now that we know that there's different microbes and we have some way of identifying them, you know, let's get some idea how different are microbes from each other. I'm telling you they're diverse. Okay, well, how diverse are they? Well, when you think about comparing two people, so you have two humans and you sequence their genomes and you compare them to each other, what's generally said, people say things like, oh, there's, you know, roughly 99.9% is identical. Humans are generally pretty similar to each other. There's only a handful of, or not a handful, but compared to the immense size of our genome, there's quite a bit of correspondence between individuals. Now, this is not true for microbes. So I'm going to show you here a plot comparing the genomes of a collection of microbes, which all belong to the same species. So in this case, Yersinia pestis, which is the causative agent of plague, bubonic plague. So each row here is a different individual. So you can think of this as being one single cell, which has been isolated and grown and had its genome sequenced. These are the sequences laid out from left to right, going from position 0 up to 4.6 million. So this is a genome which contains 4.6 million nucleotides. Compared to humans are more in the billions. So it's definitely smaller than humans. And the way that we're comparing them is by coloring the regions of the genome that tend to stay together. So if you can see my cursor on the right here, you can see at the very end, we have this yellow block. This is a chunk of the genome. And this yellow, this particular section is found in the same place. So if you follow this yellow line down all the genomes, you can see that it's generally in the same place across all these genomes. This is not true for all of them. So when we compare all these different blocks, these different regions of the genome, we can find that between one isolate and another, there may be entire regions which are rearranged and shuffled around. Now, there's this enormous amount of diversity means that depending on the species, if you compare a single individual of one microbe species, you compare two against each other, they might only share as much as 70% of their genome or 80% of their genome. So while two humans would be 99.9% identical, two microbes might only be 70% identical, even if we're calling them the same species. And I'm only sort of harping on this to really emphasize that microbes at the genetic level are incredibly diverse on a scale which is much, much larger than what we're used to seeing with eukaryotic, with plant and animal life. So with this sort of framework of there being a large amount of diversity, I now want to give you just a couple of the highlights of what we've learned about the microbiome, sort of the headlines. And then once I've given you a little bit of an idea of sort of some highlights from the field, I then want to transition to talk about how we go about analyzing this genomic data. So this is an incredibly rich data type, which also comes with its challenges. It's difficult to analyze complex data. And I want to talk a little bit about how this field approaches the challenges of data analysis and how changing technology has helped with that. And it's changing the way that we organize ourselves as scientists. So let's talk about what we know about the microbiome. So one thing I think that's important to emphasize is that the tree of life is much larger than we originally thought. And so I want to bring us back to this particular tree that I laid out before. And I want to point out this entire purple wedge here on the right. This is a group of organisms that have been named candidate phyla radiation. And these are organisms that we didn't know existed before we had genome sequencing. So there's been a set of discoveries by Dr. Gillian Banfield's group at University of California, Berkeley, where they and other colleagues were able to use these genome sequencers and apply them to different environmental and host-associated samples and be able to sequence a large number of genomes from these organisms that, as you can see, everything with a red dot on this plot has not been isolated in culture. So let's think about this a little bit. Starting with Pasteur, starting with the origins of bacteriology, we were able to study microbes when we were able to grow them in the lab. Since they're too small to physically manipulate individually, the best tool, the best approach that we've had for studying what a microbe is has only been possible by taking that and being able to grow it in isolation on a Petri dish. Now, what if that Petri dish doesn't have the nutrients for a microbe to grow? Well, then it'll just die and it'll be invisible. Only with genome sequencing has this large amount of previously invisible life become visible to us. And this is really evidenced by when you look across this tree of life and you see all the red dots, you can see there's all these organisms that we didn't know existed before the advent of genome sequencing and modern genomic analysis for the microbiome. And this is something that is very important, not only from the aspect of understanding our biology, but I think also from the standpoint of understanding how human health is influenced by the microbiome. We're not able to identify potential, good and bad microbes until we're able to actually, visualize them. We're able to measure them. So this is one thing that I think is really key when it comes to understanding the immense diversity of the microbiome, is that there are many organisms that we didn't even know existed before we started doing this type of work. Another point that I think is worthwhile to mention is that individuals are unique. And here individuals, I mean humans. Here I mean the people who are listening to these words. Your microbiome is unique. It is unique on the basis of which combinations of bacteria and viruses and archaea and fungi you have, but also the individual lineages of those microbes are different from person to person. So most people have E. coli in their gut. It's a normal, healthy bacterium. However, the E. coli in my gut is not the same as the E. coli in your gut. They have been isolated from each other by living on our microbiomes. And as they've been isolated, they've evolved independently. So an individual has a microbiome which is unique and changes at a slow rate. So my E. coli might get replaced by another E. coli from something I eat only on the time course of weeks or months or even years. So the uniqueness of your microbiome changes only slowly. Which I think is a really key part of understanding why it is that all of our microbiomes can be so different. And it's partially because we are unique. And so just to show a little bit of data, this has been observed many times. This is one publication that was part of the Human Microbiome Project, which is a large NIH-funded effort to enhance the study of the microbiome. And so on this plot, what we're measuring is how different two different microbiome samples are. So higher numbers mean that there's different sets of strains and lower numbers mean that they're more similar. So in the blue bars, we can see that if we take a microbiome specimen from one person and from another person and we compare them, they're going to be relatively different. In contrast, if we take a microbiome sample from one person and then we wait, and then we take another microbiome sample from the same person, those are the green bars, and those are much more similar to each other. This measurement is something that has only been able to be done recently, but the findings that come from this study, as well as a number of others, are really striking. The microbiome that we have is more similar to our microbiome in the future than it is to other people's microbiomes. So these green bars are lower than the blue bars, and that's true across many different body sites. And I think this is something that is really exciting. People are unique. People have different microbiomes, and there's an incredible amount of diversity there. So this is, I think, also important because what's in your microbiome influences your health. So depending on which microbes are present in your microbiome, that community will have a different impact on your health or disease compared to having other microbes present. So one example comes from this sort of dietary health. So this has to do with what happens when you eat foods that are rich in choline or carnitine. So this is food coming from, for example, eggs, cheese, and meat. So these sort of rich foods, when you eat them, they contain a large amount of higher amounts of choline, just one particular nutrient. And in the gut, choline can be changed by our gut flora into this thing called trimethylamine, which ends up getting metabolized and has these downstream negative health impacts on the heart and kidneys and atherosclerosis. Now, I don't mean to be, this is not to sort of have a dire warning. This is not always going to happen, but the negative effect of choline on our health is mediated by the microbes in our gut. They are conducting part of the transformation of nutrients, which end up producing these negative outcomes. And crucially, not everybody's microbiome does this. So only if you have the particular gut bacteria will this transformation happen. Otherwise, it's not transformed, and choline doesn't end up having this negative health outcome. People have worked on identifying which particular microbes are responsible for this. And this is largely out of Stanley Hazen's lab. And they've identified a number of other microbes where we can find that, for example, with some microbes, when you have more of this particular type of bacterium, then you also produce more of this TMAO. In contrast, when you have more of other types of bacterium, so this is sort of the name of a bacterial family, when you have more of it, you produce less TMAO. And so depending on which bacteria are in your gut, you will have the effect of this dietary input on your health will be different. And so this gives us a map of how we might one day be able to manipulate the gut flora so as to prevent these types of diseases. And that's actually sort of a larger theme that I think is one of the most exciting parts of the microbiome field, that we might be able to identify how the microbiome is impacting health, use that to design an intervention, a therapeutic, which then is able to target gut bacteria, gut microbes or other microbes in the microbiome in order to have a positive impact on human health. And the exciting thing about how early we are in this field is that there are many more diseases where we know that the microbiome has an influence, and we don't yet know which microbe. So these are things like inflammatory bowel disease, Clostridium difficile associated infection, even the degree to which cancer therapeutics are effective. We know that these processes are influenced by the gut microbiome, but we don't yet know which microbe or what mechanism. And this means that there's so much potential for discovery. We have yet to discover which microbe is going to be able to get paired with immune checkpoint inhibitors to make a more effective cancer treatment. And that's one of the reasons that I like working in this field so much. There's so much discovery ahead of us that I really believe is going to have a profound impact on human health. So now I want to switch gears a little bit, and I want to talk about data. So I'm a computational biologist. I work with data all the time. And I think that, and the point I want to try to get across here is really that there is a huge complexity to the data, but also that the ways in which we work with data have been changing in this field. And that's changed not only the insights we get from our experiments, but also the way that we organize ourselves as scientists, the way that we sort of assign job roles and communicate with each other in order to generate these analyses, in order to conduct these experiments. So the first thing to mention is that microbial data analysis is complex. This maybe goes without saying, but I've talked about a microbial genome in a really abstract sense. I've talked about genomes being different, and I've showed you some figures. Now I want to show you what these genomes actually look like if you were just to open up a file. So on my computer, I have a bunch of different files with different microbial genomes in them, and I picked one at random, and I said, okay, let's open it up here and show people what it looks like. This is actually what the genome sequence is. So I told you that a genome is just a word of A's, T's, C's, and G's laid out from end to end, and there are a couple million letters long for a bacterium. Well, this is that. This is exactly that. So this is the Bacillus cereus genome, this particular strain. And here's the first nucleotide in the entire genome. You read across the line. This is the first 45 or so lines of that genome. And it goes on for a few million letters. So this is an incredibly rich piece of data. There's a lot of information in here, and I can't read any of it. I don't think that almost any human can look at a genome sequence and get an understanding of what it means. Okay, so it's a complex data type in the sense that the raw data, the A's, T's, C's, and G's contain a lot of meaning, but it's hard to bring it to the human understanding. And so one thing that scientists spend a fair amount of time on is developing tools for taking these complex inputs. So for example, a collection of genomes, and then figuring out how to compare them to each other. So this is a visualization. This is actually a type of visualization, which is produced by a software tool called Anvio. This is a really wonderful piece of software in the microbial space. It's produced by Murat Iran's group in the University of Chicago, who's a fantastic scientist. And what this group has done has made a program which can transform those files I just showed you into these interactive displays. So in this display, every concentric circle is a different genome. There's 31 of them in this analysis. Whenever you see a dark line radiating outwards, that means that there is a gene that is found in common between these genomes. So there's a wedge here of genes that are found in all 31. Here's a wedge of genes that are only found in this sort of bottom half of genomes. Here's a wedge of genes that are found in sort of this middle section of genomes. You can do all sorts of things with this display. So this is a very, very rich representation of how to compare these microbes. And you can use this to group together different genomes using this tree I'm showing you here. We can also infer the function of each of these genes, so each of these wedges along the entire thing, and then add many different annotations. I'm not going to try to tell you exactly what this display means, but I just want to bring it up as a demonstration of how software can transform raw data into a representation that can provide insight to a scientist. So the scientist who did this experiment would be able to explore this visualization and come to the answer of, here is the biological answer to the question that I had. Here's the discovery that I was looking to make. So great, I told you data is complex, and I told you that there are these tools that can make them visually accessible, that can make them interactively accessible. Well, unfortunately, they're also very difficult to run. So what I'm showing you here is one of the commands that you would need to execute in order to transform these raw files into this interactive display. So this is a command that you would type in on the command line. So if you're opening up a terminal and you're interacting with your computer, not with a mouse and windows, but instead just by typing text and reading the text that the computer gives back to you in response. And so in order to generate this display, you'd have to install the Anvio software and then type in this command with all these different flags or options set in the correct way so as to produce the output that you desire, that you require. And I think the point here is that it's difficult to install and run the scientific software that's used to produce really informative outputs. And that is in no way a criticism of the scientists that produce these tools. It is just honestly hard to make software that will run across many different computers. Installing software is hard. That's why we have these whole industries of software development. And so when you have an academic who's making a software tool, they're able to do something that has lots of scientific value, but they don't have the resources of a Microsoft behind them in order to make a distributable piece of software that you can just double click and install. And so this means that the traditional model for how data analysis is conducted in an organizational sense in sort of these data intensive research communities is that you have some set of input data and then it's processed by someone like me. So someone who is trained on the command line would then know how to install and run this Anvio tool, which then produces an interactive visualization. And then I would take that interactive visualization and I would try to give it to the researcher I'm working with so that they can come up with their own insight. So a biologist, given that there are many more people who are interested in consuming this data, but they're not necessarily trained in all the nuances of how to install and run all the different pieces of software that you might need. And so this organization, it's very simplified, but I hope you can appreciate how this can cause a bit of a bottleneck. So when a researcher wants to ask a new question, they have to put the data through this pipeline where it has to go through someone like me in order to analyze it and give them the answer they're looking for. This is not a huge problem, but it slows the pace of research. It slows the time it takes from idea to answer, from insight to sort of realization. It adds that gap of time and just slows the entire process. Now, in the theme of how technology is changing the way we do science, this is a perspective piece from Nature from late 2019, where they talk about an entirely new type of software called workflow managers, these computational pipelines. And what they were trying to emphasize here in this article is that instead of making specific tools, a few different groups have been making sort of a flexible, open-ended piece of software, which can wrap around and run any other piece of software. So it's a little bit meta. It's a little bit of a sort of like, Russian nesting dolls here. But the idea is that you have a piece of software that's really good at running other people's software. And the reason that that's convenient, so it sounds like maybe an added level of complexity, but it means that for a user, all you have to know how to do is run one piece of software, the workflow manager. And if you know how to run this central piece of software, then you're able to wrap around and execute analyses using any of these individual scientist-developed visualizations or analysis workflows. So just to try to put this in the context of the same sort of flow diagram I had before, when you have access to something like a workflow manager, then I, as a collaborating computational biologist, I can make a workflow which runs Anvio. And this workflow will take input data and produce a visualization. Now, instead of me running the data myself, I am then able to train a scientist who is not typically focusing on computational analysis. I can train them how to run this workflow manager. And with that skill, with that technology, this researcher is now able to run their own analyses. They can pick which data they want to analyze. They can access the interactive visualizations. They can consume it themselves. And this takes me out of the loop. So the trained computational biologist, all that they are then need to do is know how to make these workflows and train researchers on how to use workflows in general. And that puts the power into the hands of the scientists, where then they are able to analyze their own batches of data without having to put the data directly in my hands. Now, there's a specific example of this that I wanted to highlight, which was I was working with one microbial researcher in VID, Chris Johnston, was interested in querying a number of genomes, a number of bacteria, and identifying when you have groups of genes that are found together. So here we have each line is a different organism. And he was really interested in finding when you had this green gene next to this teal gene, which is next to this blue gene. So this pattern of 1, 2, 3, these are right next to each other. And wanting to pick out, okay, here's a genome where they're next to each other. Here's one, here's one. Down at the bottom, it's actually flipped in the opposite orientation, but the relative position is the same. And it was this pattern that he was really interested in being able to pick out, being able to search for operons surrounding any gene of interest. Now, instead of just doing the analysis for him, what I ended up doing was making a tool. So I made a workflow for, it was a bacterial operon finder for functional organization, or BOFO. And well, all I had to do was build a workflow and then train researchers in the Johnson lab how to run this workflow. And then all of a sudden, they were able to analyze genomes themselves. And this setup was actually ended up being incredibly convenient because the first time I made this tool, it wasn't done. I got Chris gave me the idea of what he wanted. I put together a workflow. It got to the answer and he looked at it and said, it's not quite right, Sam. We want to add this, we want to add this particular nuance. We want to fix this bug. And instead of my having to go in and fix each bug or add each new feature, all I had to do was update the workflow. And then that software would automatically be distributed into the hands of every researcher who wanted it, and they'd be able to analyze their own data themselves. And so in that way, this sort of bipartite organization being centered around this new technology has supported more rapid innovation of methods and innovation of science in a way that would have been really difficult to do using any other sort of analysis technology. Now I'm going to take this all one step further as I'm wrapping up with this section here and talk about how this approach has allowed us to reorganize the way that we analyze data for microbial research. So the idea here is that as a computational biologist, I'm able to focus time on individual workflows. A workflow that finds operons, a workflow that runs ANVIO, as well as a number of other workflows that are made by the community. Every single one of these things are compatible with a single workflow manager called Nextflow. And so this means that every single researcher out there who's trained in how to run a workflow can now select each of these different tools from their toolbox and pick which one they want to run on their data. And so this effectively ends up multiplying the impact of every single computational biologist, because when I put time into an analysis, into developing analysis, it's not just a single analysis that's run once. This is analysis that can be run many times by any researcher in the sort of free and open source software community. And the other thing you'll notice here is that I am no longer the bottleneck of analysis. As a computational person in the past, I've often felt frustrated that in order for a scientist to analyze the data from their own experiment, they would have to wait for me to spend the time to type in commands at the command line. That's something that I know ends up slowing the pace of science. So with this, by implementing the right technology, we're then able to change the way we organize ourselves as scientists and put more tools into the hands of researchers. Hopefully this is not necessarily something to get you excited about workflow management tools. I think those are very specific to our field. They're very specific to scientists who are analyzing large amounts of data. Instead, the aspect of this that I want to emphasize for the broader audience that's listening right now is that there are ways to invest in technology which end up reorganizing the way that we arrange ourselves as a group of people trying to get a task done. And so this ends up creating a new type of job role. So instead of being attached to a single lab, as part of the Microbiome Research Initiative, I'm able to support a number of different labs, a collection of labs, many more than I'd be able to support if I were doing this sort of individual analyses. And hopefully there's a similar type of lesson to be learned from the larger sort of administrative support world. Where sometimes the best thing is to perform a task on behalf of someone, and sometimes it's best to develop a tool which you can put into their hands so they can perform that task best themselves. It's always hard to know when to make that trade-off, but I think this is one example when putting tools into the hands of researchers ends up being much more efficient for everyone. So I hope that you enjoyed a little bit of discussion of core technologies, and I do want to make sure to emphasize a little bit of the science of the microbiome that brought us all here, and the science that I think is really quite exciting. So just to reiterate, when we think about the microbiome, we're talking about all the microbes living on the body. And this contains hundreds or thousands of different species, mostly bacteria, lots of viruses infecting those bacteria, also microeukaryotes, so fungi, also archaea, and so these novel forms of life. Our ability to understand the microbiome and to measure what is present in the microbiome is something that was completely revolutionized within the last 20 years by the advent of high throughput genome sequencing technology. So as a result of the investment that our society made in the development of genome sequencing, that has opened up an entire new field of research, which I believe is going to have a profoundly positive impact for human health. As a result of this new technology, we have discovered many more microbes than we realized existed before we did this. This is entirely new clades of the tree of life. Our understanding of how life evolved on this planet has changed dramatically as a result of this new data that's being generated. Now, how it impacts you as an individual, you have a unique microbiome. The combination of bacteria, of microbes that are present in your gut is something that only changes slowly over time, and that makes you distinct from every other person. This is something that I think is really important and also impacts human health. There are so many things that we know the microbiome is related to. We have so many ways that we know the microbiome influences human health overall. And for most of those, we do not know yet what the exact mechanism or reagents are that can perform that manipulation. And so what I'm talking about here is developing therapeutics. I think that the world of microbiome-based therapeutics is a large one that has only started to become explored. And that in our lifetimes, I think we have a lot to look forward to for the promise that this expansive new field of research has for the impact on human health and the treatment of major diseases. So those are the materials that I had prepared for you today. I hope that you enjoyed this, and I'd be very much happy to take any questions and continue further. Thank you.
Thanks very much for presenting this morning, Sam. It's always great to hear about our research going on at Fred Hutch. Let's see what we've got here for questions. Okay, so the first question, what do you see as limitations in the current workflow manager that will likely be improved upon in the near future?
This is something I spend a lot of time thinking about. So, how to improve and deliver the analyses that are performed by workflow managers, really as a way of performing analyses, any sort of high throughput analysis. And I think the core goal is, how do we put a tool in the hand of a researcher who's then able to analyze whatever their data is using whatever resources are available. So right now at the Fred Hutch, we have an amazing high performance computing system. We have a SORM, we have these networks of computers which are able to perform an amazing amount of computation. And right now we've gotten it as simple as a researcher being able to execute a single command on the terminal, which is able to then marshal these resources and direct them to their data. I think there are ways to improve this. For example, there are graphical interfaces where instead of ever opening up a terminal, a user might be able to open up a website, which would then be able to show them the analyses that they can perform for their lab, and then click through some menus and be able to perform really powerful computing, but from the sort of comfort and ease of something where you can just sort of point and click. And that's something that I think is very much possible, and I'm looking forward to being able to help implement here.
Excellent. Do we have any other questions? Looks like that's the only one that has come in so far.
Okay.
Let's see, there's about 10 minutes left, but okay, here's another one that just came through. Do therapeutics and treatments affect the microbiome? If so, can you speak to how you conduct research on people in therapy?
It is absolutely the case that therapeutics affect and are affected by the microbiome. In a lot of ways, this is an area that we're only starting to understand. I'd say that the areas of research here that are particularly informative. So maybe if I can provide one example that might be of interest to the Fred Hutch community, I would focus on cancer therapeutics. So there's, as you may know, there's an entirely new class of cancer therapeutics called these immunomodulatory. So these like immune checkpoint inhibitors are one of them. So cancer immunotherapy is something that is obviously expanding rapidly. It's very effective. And then some of these involve things like CAR T cells, so cell-based therapies, and some of them are small molecules or these sort of immunotherapies. Now, what people in the microbiome field have been starting to observe is that the effectiveness of particular immunotherapies depends on what microbes are present in the gut. And that might seem like a bit of a stretch, like, oh, that's so implausible. How would a cancer therapy depend on what's in your gut? And I think that approaching the topic with skepticism is the right way. However, there is a direct connection, which I think explains this rather well and is rather intriguing, which is that of the human immune system. When you think about microbes in the gut, they are physically adjacent to a large amount of human tissue, the lining of the intestine, which is very much in contact with the immune system. So the immune system is monitoring and interacting with the contents of our intestine, which means that there's only a single step for the contents of the microbiome to influence the amount of sort of inflammation or activity of the human immune system, which means that it's better or worse prepared for responding to the immunotherapy. And I think that this is one area that's proving to be one of the most exciting, is the way that the microbiome influences the human immune system. And there's the mechanisms for how this is carried out are only starting to become understood. And the potential here, when we think about doing our own research, is that by understanding how the microbiome influences the immune system and influences the response to cancer immunotherapies, the goal is that we as researchers would be able to identify particular microbes which could be given alongside an immunotherapy in order to provide a more effective cancer therapeutic. That's an example of just some very early research that's being done here, as well as labs around the country and the world.
Great, thanks. Next question. Are there any publicly available workflows and data sets that you would recommend for introducing a new scientist to using big data?
Absolutely. I think one of the great things about using workflows to analyze big data is that you're able to use workflows generated by anyone. So the goal here with using workflows is that a workflow developed in London could be used by a researcher at Fred Hutch with only minimal need for sort of reconfiguration. The community, there's a number of different communities of scientists around the world who make these large data analysis workflows. And one of the ones that I'd like to highlight is called NF Core. So NF Core is a group of core lab scientists around the world who have centered around the use of a particular workflow manager called Nextflow in order to run all sorts of analyses, RNA-seq, ChIP-seq, lots of different next-gen sequencing-based analyses. Now, in order to run those workflows on our infrastructure, all you have to do is configure Nextflow to run in your account here at Fred Hutch. We have documentation, we train lab members, and that's something that I do quite regularly. And so I'd say for anyone who's interested in this sort of large-scale data analysis, that's very much my role, and my continuing role here at the Hutch will be helping scientists utilize these workflows on their own data. So I'd encourage anyone, feel free to get in touch on this topic.
Fabulous. Okay, there's one more question here. Do you foresee microbiome analysis getting to the point where patient microbiomes can be readily sequenced and binned to assist with personalized medicine? Are there particular diseases where this would be most feasible?
I absolutely see this as part of the future. I think one of maybe the simplest example that you can think of is, for example, this atherosclerosis. So eating the red meats and cheeses, example that I gave earlier. As technology improves, it's going to become more and more practical to do something like sample a person's microbiome and identify whether or not they would be at risk of heart disease by eating red meats and cheeses. So by measuring the microbiome, we should be able to assess relative risk. And also what types of therapeutics might be helpful, microbiome therapeutic-based therapeutics or other ones. I don't think we live in that world yet. The technology now is not necessarily as rapid or as low cost as would be needed in order to transition a microbiome-based diagnostic for any sort of research activity into the clinic. However, it has already started. So this process is possible, and it does form the basis of some diagnostic tests, even ones developed by researchers here at Fred Hutch, based on the composition of the microbiome. And I believe that that'll only expand as our knowledge increases of which particular microbes are involved, and also as our technology for characterizing the microbiome in a point of care setting, as that also improves.
How far out do you think that would be to have more diagnostics in place?
I think that in terms of genome sequencing, we exist at the cusp of an inflection point, I believe. So genome sequencing technology has accelerated rapidly with Illumina sequencing, but the basic technology being used has only been improved incrementally over recent years. Sorry, this is my personal opinion. This is not a criticism of any sort of commercial entity in any way. But there's a number of new technologies for genome sequencing that use completely novel molecular approaches. So without going into depth, there are new technologies that would transform the genome sequencer that you've put on a bench right now into something that fits in the palm of your hand. And those are continuing to get better and better. And at some point, I think in the not too distant future, these extremely small genome sequencers are going to start to become superior to the large-scale benchtop genome sequencers. And when that happens, then all of a sudden, I think we're going to see a turnover and many, many more applications for mobile, diagnostic, point-of-care, genome-based technologies will start to become possible.
Yep, that makes sense. Great. Okay, well, it looks like that's our last question, and we're just at about the hour. So Sam, on behalf of Hutch Connect and the Science for Staff committees, thank you very much for presenting this morning. I'd also like to say thank you to Lex Lamug, who helps record these seminars, and she helps me organize them, every single one that we've been doing virtually. So thanks again, Sam, and thank you to everyone who joined us today. Everyone have a great afternoon.
Great, thanks to Hutch Connect, and thanks to everyone for listening today.